Hello, my name is Jason Adrians. Today is 4/18/25, and this is my Unit 3 presentation. So after reading the article by Bernardi, to answer the question, what exactly is the non-coding DNA mystery, you have to understand what DNA does originally. And what DNA does is essentially the central dogma of molecular biology. DNA codes for RNA, RNA codes for proteins. So it can be thought of as DNA makes proteins, essentially. And how DNA makes protein is through things called genes. So there's promoter regions on DNA, which it's why it's called the promoter region is because RNA wants to go to that region. And then RNA reads that DNA, RNA polymerase reads that DNA, and then makes RNA from that DNA code. The problem with this, or what the non-coding DNA mystery is that nearly 98% of DNA actually doesn't code for anything. So nearly all of DNA actually doesn't do anything. So that's what the mystery has been. What is the purpose of this 98% of DNA that actually doesn't make any proteins? And what Bernardi hypothesized and ultimately came to the conclusion was that this non-coding DNA actually makes up how the DNA folds, or how the chromatin basically folds on itself, because if you didn't know, the DNA molecule is very long, and in order for it to fit into a nucleus, it has to fold on itself multiple times. And why this is because DNA has regions in the non-coding DNA parts. It has regions called isochors that have a high guanine and cytosine rich area. So in order to abbreviate this, I'm going to say the GC rich region and a GC poor region. So in a GC rich region, the DNA actually doesn't fold on itself a lot. And in a GC poor region, the DNA folds on itself a lot. And why this matters is because, as I mentioned before, RNA polymerase wants to go to a promoter region on that DNA. And if the DNA is folding a lot in that region, it's hard for RNA polymerase to attach to that DNA there. So what Bernardi basically concluded, and what concluded with his evidence data and from what he hypothesized, is that there's actually two types of codes in DNA. There's the DNA that codes for actual proteins, and then there's what he called the genomic code, which is how the DNA folds on itself ultimately to get, it basically is an inhibitor and a promoter of what proteins get made. If, for example, if there's an area on that DNA that's GC poor, guanine and cytosine poor, there's not a lot of proteins that are going to be made if there's a gene in that specific area, because it's going to be tough for RNA polymerase to get in that area of the DNA where it's folded a lot, versus if there's a GC rich region where a lot of genes are and its DNA is not folded a lot, those proteins are going to be made a lot more frequently. So that's basically what he concluded from this article. In my personal opinion on how this article advances our understanding of the non-coding DNA mystery, I think it advances genetics quite a bit. Previously, there's been 90, people have wondered what does this 98% of DNA do, and I think that question has been answered. It essentially acts as a secondary or a primary promoter versus an inhibitor of what proteins get made versus not. So that's just a great explanation of what he stated in this article. And yeah, thanks for listening.